Well, good morning, everyone. And first of all, thank you for the opportunity. So it's great to have a community of glycans, and galectins. So I'm an immunologist. Obviously, first of all, I would like to, because I talk about the immune cells. So obviously, we have two arms of immune system: innate immune cells. We have macrophages, neutrophils, and monocyte. There are other cells, but I don't talk about them. And then we have some cells that they are bridging the innate and adaptive immune cells. And in the adaptive part, we have two main cell population, T cells. We call them CD3. And they will be divided in two main groups of T cells, CD4, that we call them helper T cells, and CD8 or killer T cells. And we also have B cells that they are secreting antibodies. So this is just a kind of background that I will talk about them. So the previous talk was helpful because covered some of my introduction. So it just gives me more time. But the main thing that I just want to confirm the previous talk, that obviously galectins can bind to non-glycans. And especially there are some reports that inside the cytosol, they can interact with non-glycans. And we have 15 galectins so far have been identified, and they have a wide range of biological functions. But galectins, they don't have a transmembrane domain except galectin 3. And they are everywhere. So inside the immune cells, non-immune cells, in the cytosol, in the nuclei, and even in the cell membrane. So they are highly abundant, and they have a diverse kind of biological role. And in terms of concentration, in terms of expression level, it just varies depending on the cell, depending on the kind of microenvironment that they are present. This is just repeating what presented in the previous talk. Obviously, galectins, the main common area that they have, like in terms of structure, they have this CRD, and they can interact with the glycans, and they have very high affinity for beta-galactosides. However, they can bind with non-glycan components as well. And in terms of structure, obviously, there are three groups. And you can see that, like a prototype, we can see them as monomer, as dimers, but galectin 3, which is the chimera type, we can see as monomer, dimer, pentamer, N-type polymerization, and C-type. And a few of galectins, such as galectin 4, 6, 8, 9, and 12, they have a kind of tandem repeat. So these CRDs are with a peptide linker. They binding together. So it means that for example, this galectin can, with two CRDs, can interact with two different receptors at the same time. And that makes it very complex when it comes to biology, because different cells can secrete these galectins. So how they are secreted, we don't know. I mean, this is a non-classical pathway. They get released from the cells. And when they are released, obviously, they can interact on the surface of the same cell. So they can interact with the glycoprotein receptors, and they can interact with the extracellular matrices. They can be involved in cell adhesion, for example, galectin 9, galectin 8. They have two CRDs. They can bind the two cells together. They can form lattices, and obviously this interaction, depending on the nature of those receptors, results in a different signaling pathway. And they have a diverse role, biological roles in terms of development and in terms of immunomodulatory function. When it comes to cancer, it's very controversial, but there are, if you look at in the literature, there are lots of papers that they emphasize that GALs are involved in metastasis and tumor invasiveness, and they are involved in immune cell exhaustion, T cell apoptosis, but there are some reports that showing that galectins are helping in the context of cancer because they induce apoptosis in tumor cells. So it's very controversial. But in terms of interaction, so in the past, we have been kind of told that galectins only binds to self-glycans. But recent evidence shows that galectins also bind to non-self glycans. It means that they can bind to glycans on the surface of pathogens, worms, like parasites. They can bind to viruses, bacteria. And so it means that depending on the nature, they can either increase or inhibit infection via this interaction. But another interesting concept is that galectins can function as damps, damage-associated molecular patterns. Because in biology, we have PAMPs, pathogen-associated molecular patterns. This means that our innate immune cells recognizes different components of pathogens, like, for example, LPS, flagellin. Those are PAMPs. So when they recognize them, they become activated. And immune cells also recognize DAMPs, and galectins function as DAMPs, so that immune cells recognize them that there is a damage, and it results in activation of a variety of immune cells. But obviously, galectin, as I said, there are 15 of them, so there is no time to talk on them. So I just focus on one of GALs, which is the most studied GAL, which is galectin-9. It's the most studied GALs, and just imagine, galectin-9 can interact with at least 9 receptors. So when it comes to biology, it becomes so fascinating and sophisticated. Okay, why galectin-9 likes to interact with TIM3 instead of PD-1 instead of PDI and blah, blah. So it's very hard. So we don't know the affinity, if a GAL has differential affinity for each of these receptors. So these receptors can be expressed on one single cells or different cells. So it makes it very, very difficult to study how galectin functions, because they interact with different types of receptors. So I start my talk, kind of my journey with galectin-9. So it started a long time ago. So in 2008, when I went to University of Washington to do my second postdoc, so I was asked to answer this question. Why a subpopulation of HIV-infected individuals have natural immunity? So there are a small population of HIV-infected individuals. They get infected with HIV, but they do not progress to AIDS, even if they don't take treatment. So they have natural immunity. They can maintain low viral load. Sometimes it's undetectable in their body. They don't have any symptoms. They are healthy. And however, they have virus in their body. So this was fascinating, that why these small groups. We call them elite controllers or long-term non-progressors. It means that they get infected for a long term, and we have patients that they have been infected for 40 years. And they are still healthy. Otherwise, I mean, they have the virus, but they don't have any symptoms, and some of them, they don't take medications. So this was fascinating, why they are kind of protected. But one thing was known at that time, that these individuals are enriched with certain HLA type, specifically HLA-B27 and HLA-B57. And we know that HLAs are very important in terms of antigen presentation. So antigen, as you remember, on those bridging cells, dendritic cells, they process antigen, and then they bind it to MHC, which is in mice, we call it MHC, in human HLA, and then they present it to T cells. And those MHC class 1 or HLA class 1 are very important in terms of antigen presentation. And those individuals, obviously, that are enriched, or they have HLA-B27, 57, so that means that there is something with these HLA that provides protection in this individual. So I was asked to answer this question. So I worked 3 years, very long days, including weekends. So this is for graduate student postdocs. You have to work beyond working hours. So after 3 years, I make it in one slide, what I achieved. So if you look at those individuals that have HLA non-B2757, like, for example, HLA A02, A03, B08, B07, there are so many of them. What you see that this CD8 T cells recognizes an infected cells. So helper T cells are the main target for HIV. So HIV infects helper T cell. When this CD8 T cell, which is the killer cell, recognizes on this MHC class 1, which is non-B2757, and this is the antigen little one is presented on this MHC, and then this is the T cell receptor recognizes that this cell is infected. And those that are restricted by non-B2757, when they see antigen, they upregulate tons of TIM3. And then a subset of immune cells, we call them regulatory T cells or Tregs. They are like policing the immune system. Their job is immune regulation. So if the immune cells become highly activated, they tell them no, stop, because you can cause collateral damage. So these are very important in immune homeostasis. And we found that these regulatory T cells express galectin-9. And then the interaction of galectin-9 with TIM3 results in a negative signal. So it means that turning these CD8 T cells off, they're making them, we call this term of exhausted. They become exhausted, impaired, they are unable to fight back. And then what happens that we see viral replication, we see increased virus load, and then the virus wins in this scenario. But when you look at the B2757 on the other side, that was very fascinating that when CD8 T cell restricted by HLA B2757, when they see infected cells, the same mechanism, they do not upregulate TIM3. Very little to none. And therefore, they cannot be suppressed by regulatory T cells. That means they evade immune suppression. Another interesting thing that I observed that these CD8 T cells not only do not get suppressed, but they fight back and they kill Tregs. This was very fascinating. And then what happens here that they proliferate because they don't get suppressed, they proliferate, they expand, and they kill infected cells. In this scenario, a host wins. So another study that I did at the same time, I was just curious to see that that was the surface-bound galectin-9. So how about the soluble galectin-9 when it's secreted? I was just curious to see that whether soluble galectin-9 has any effect on HIV infection. These are target cells of CD4. So usually we activate T cells because the virus relies on the cell machinery of the host. So we have to activate them, stimulate them to become more appealing, more attractive for the virus. And then when you activate T cells, CD4 T cells, they upregulate TIM3. When you add galectin, recombinant galectin to them, the same as previous slide, the interaction of galectin and TIM3 results in an inhibitory, a negative signal. So what happens that CD4 T cell, they upregulate protein P21, or P21 or protein 21. The upregulation of protein 21 results in downregulation of cell cycle machinery, and the virus relies on the cell machinery, so it cannot replicate. So we see that the interaction of galectin-9, soluble galectin-9 with TIM3 results in reduction or resistance of CD4 T cells to infection. However, if you do the same assay on rested CD4 T cells, you don't activate them. It means that they don't have TIM3. If you do it, you see that the opposite effect. So you see that galectin-9, when it interacts with this CD4 T cell, the infection goes up. So we found that this happens through another receptor, which is protein disulfide isomerase. So what happens that galectin-9 interacts with PDI, and it retains PDI on the surface of the cell. And this retention results in the interaction of PDI with the CD4, which is the main receptor for the HIV, and also CXCR4, which is a co-receptor for HIV. and then it's interact with GP120 of the virus, and then it hijacks the virus and it brings it inside the cells. So as you can see that how complex it is. One galectin, one cell type, depending on the status of cells, have completely opposing effects. And at that time, we published it in Blood, and at the same time, another group published the same, but they didn't look at, on the other side, just this part they published in PNAS almost a decade ago. And then I started looking, when I have my lab here, I started looking at the expression of GAL on variety of immune cells. These are T cells, obviously CD4, CD8. So we started looking at the expression of galectin-9 just in HIV patients versus healthy individuals. So this is a flow cytometry plot. Obviously, each dot represents one single cell. And you can see that this is the healthy individuals. In healthy individuals, we don't see much galectin-9 on the surface of T cells. However, when you look at HIV-infected individual, you see massive expansion of galectin-positive T cells. And then we wanted to see that what is, in terms of function, difference between those galectin-positive and galectin-negative T cells. And we did extensive work, but I just present one or two slides. And then we look at their function. So we look at 3 cytokines. So in this quadrant, you can see that these are galectin-positive cells in this quadrant, and those are galectin-negative. And this is unstimulated cells. Obviously, you shouldn't see much cytokines, so interferon gamma, TNF, IL-2. But when you stimulate them, you see that only galectin-negative cells secrete cytokine on the other, in this quadrant. But in the quadrant, this galectin-positive, almost they have 0. So that means that they are unable to secrete when they have galectin on their surface. Also, we look at proliferation capacity of those gal positive and negative. As you can see that they have significantly lower proliferation capacity. And also, we look at this perforin and granzyme. These are cytolytic molecules. So killer cells secrete granzyme and perforin in order to kill infected cells or tumor cells. And you can see that the gal positive cells also significantly have low level of these molecules. And then we wanted to see that, okay, what is the partner of galectin-9? So we found that this is just imaging, this is just the controls, and you can see that here there is lots of co-expression of galectin-9 with CD3, which is the T cell. And then we see that galectin-9 interacts with CD44. And we found that PKC results in the activation of T cells. But you see that they don't have any cytokine. But it initially results in activation. But prolonged activation of immune cell results in exhaustion. They become impaired. They don't have any functional properties. And then we also look at other molecules. This was kind of interesting. So there are lots of things can be done in the future. So you see that these are all inhibitory molecules. On the surface of T cells, we can see them. But what was fascinating that all the PD-1 positive were GAL positive. We did imaging and we saw that there are some co-expression, but we didn't dig into it further. We published in paper in 2020, and 1 year, another group published that they did further investigation, and they found that galectin-9 interacts with PD-1. So that means that one CRD interacts with TIM3, another CRD with PD-1. So this shows that how interesting galectins are. And just a summary of what I showed, that GAL interacts with TCR and CD44. And then initially, obviously, results in activation, but persistent activation results in downregulation of all of these and exhaustion of cells. And these cells were terminally exhausted. We tried different ways to reinvigorate them, but we could not. So those GAL positives are highly exhausted. And then another cell type that we were interested to look at it in HIV patients is neutrophils. So this is extensive work. I just want to show you a couple of slides. So we look at the healthy control genes. These are low CD4 and high CD4. Low CD4 means that those individuals that, because CD4s are HIV target cells, that means HIV has killed lots of their CD4, and they have gone on treatment probably late. And as you can see, these clusters, those that have lower CD4, their neutrophils are more activated. We did extensive studies, but we came to a very nice kind of observation. We found that neutrophils in healthy individuals are almost covered with galectin-9. You can see that here in this quadrant, 99% of healthy control neutrophils have galectin-9 on their membrane. And this is just a cumulative data. But HIV patients, some of them almost have zero GAL. But there is a wide range. So we wanted to see why there is a wide range. And then we found that those have low CD4, they have very low GAL. But those have high CD4, they have kind of moderate levels of galectin-9. So the question was that how they release or shed galectin-9. So this is a very large study, but we found that when neutrophils become activated, they shed GAL. So this is a stimulating neutrophil with LPS, as you can see that there is an hour stimulation is enough. You see significant decline in neutrophil. And if you just collect neutrophils and just culture them in the Petri dish, and from healthy individuals, high CD4 count, low CD4 count HIV, you can see a significant elevation of GAL shedding from neutrophils of HIV infected that have low CD4. It means that they are more progressed in terms of HIV. So this is very complex, but we found that there is a very interesting kind of mechanism is happening that results in, I don't go through it because I don't have time, but this is in the paper. So we found that GAL is in the kind of lipid draft, interact with CD44, and it through the activation, and ROS is very important in this scenario, results in rearrangement and movement of CD44, and finally galectin-9 shedding. I just summarize it here. So a couple of years ago when the SARS-CoV started, so we started looking at galectin-9 in the plasma of patients. And you can see that COVID-infected individuals, they have skyrocketing. We have never seen this level of galectin-9 in the plasma. And then when you divide patients to moderate and mild, that means those who stay at home and go in the hospital ward, and severe means that ICU admitted. So ICU admitted had significantly elevated more galectin-9 in their plasma. And then with about 2,000 picogram per ml, we were able, with 95% sensitivity and specificity, we could use as a kind of diagnostic, galectin-9 as a diagnostic marker in COVID patients. But what was striking that when we look at the, ELISA multiplex, you can see that GAL is the highest one. And then obviously lots of pro-inflammatory cytokine. But what was kind of more interesting that there was a positive correlation between lots of cytokines and chemokines with galectin-9. So we found that galectin-9 activates a lot of cells like NK cells, monocyte, and they secrete IL-6, TNF-alpha, which are inflammatory cytokines. And we did see the same story. We see that in the context of COVID, neutrophils shed galectin-9. So that means neutrophil might be a major source of galectin-9 in the context of inflammation, because neutrophils are the most abundant cells in our body. So, and those are short-lived. They live 1 or 2 days and they are dead, and another cells. So just imagine that if they're shedding galectin-9, if we have inflammation, it's going to be a huge problem. And we propose this kind of cartoon that in the context of SARS-CoV-2, because we have damage in the respiratory tract, they release galectin-9. And that galectin-9, as I said, we have pathogen-associated molecular patterns, which is the viral antigens, and then galectin function as a damp, and then these alveolar macrophages become activated, they start secreting galectin-9. And galectin-9 also can interact with other innate immune cells. They become activated, they start releasing galectin-9. And then this results in the cascade of cytokine storm. So we propose in this study that the main drive for the cytokine storm in COVID patient is galectin-9. And we have some studies looking at different types of the viral strain and variants. And as you can see that in the Wuhan, we had the highest levels of galectin-9 in the plasma. And it makes sense that cytokine storm was killing more people in the Wuhan compared with other strains. And then in the Delta, lower, and Omicron is the lowest. And then we start looking at Gal8 and Gal3 in the plasma, and we see that those are also elevated. Specifically, they are highly elevated in the Wuhan strain versus the Omicron. So Omicron had the least Gal levels. So I briefly go to the cancer part, just a couple of slides. So we had a clinical trial, phase 2 clinical trial. So we had some viral-associated cancer. These are EBV, HPV. And then, as you can see, these are cycles. So the patients went on valproic acid, and then 2 weeks after, they went on this avelumab, which is anti-PD-L1, one of the immune checkpoints. And then we collected blood, different time points. But this cycle 7 was the time that kind of patients were assessed in terms of immune response, in terms of treatment outcome. So we found that galectin-9 is highly upregulated in these cancer patients on their T cells, CD4 and CD8 T cells. And those T cells were like HIV. They are exhausted in terms of cytokine production. But we had a kind of interesting CT scan from a patient that was responder and non-responder. This is just an example. So you can see that this is the A and B, the tumor size. You can see with the scan, but as this is cycle 7, you can see that after treatment, the tumor is very small. I mean, there is a kind of partial immune response. And what was interesting, that CD8 T cells, they had very high level of GAL. And then by cycle 7, GAL was very low. And we see that this good treatment outcome. And this patient had kind of moderate galectin-9 when we started treatment, but then it went really up. And this is the case. You can see that the tumor is here, and then we can see the tumor is increasing, and even we have metastasis in the liver. So it shows that how important GAL can be. Another thing is, I mean, there are different genes, but this is the same patient. So we did biopsies, and we look at the total galectin-9 gene expression. In this patient that had partial responder, compared with the screening time, there is downregulation of galectin-9. But this one, there is no change in terms of galectin-9. And another very interesting study that we are kind of is unpublished data, so, but there are lots of things, but this is kind of very interesting. We look at the, galectin-9 expression. So these are 7 patients. These are non-responder. You can see that the galectin-9 expression versus these 4 that responded to those treatment. So it shows that galectin-9 level at the gene level can be used even in the peripheral blood as a kind of prognostic. So it just comes to the precision medicine. Instead of putting all the patients on treatment, just selecting those that potentially will respond, and then focusing on those instead of putting all the patients on treatment. So I just, this was just something that I mentioned. So I just have my general kind of conclusion or summary. So in the context of inflammation, infection, or cancer, obviously, we have lots of immune activation. Immune cells become activated because they see antigens, whether it comes from cancer, from HIV, Hep B, Hep C, anything. So they become activated. And then when immune cells become activated, we have immune cell apoptosis. So when cells go to apoptosis, they have lots of galectin. They release it. And even when tissues get damaged, they release galectin-9. So these galectin-9, they can interact with different receptors. And they can activate other cells. When the other cells become activated, they also secrete cytokines and chemokines and also galectin-9. But one of the very important cells are neutrophils, as I showed, because these are the most abundant cells. When these cells become activated, they shed lots of galectin-9. And this creates a vicious cycle. More galectin activates more immune cell activation results in the release of more galectin-9, and then it continues. So that's why that gals can have a really, I mean, especially galectin-9, others have. We have started working on, Juan is sitting here, we started working on GAL8, so, and we also started working on GAL3. So it's very kind of interesting. So with that, I would like to acknowledge some of the work that, I mean, we have done lots of work, but I just showed a few examples. So my past and present lab member that did a variety of kind of work, and my collaborators. So the HIV cohort that we did, one thing I didn't mention, I have to acknowledge that Stan Houston, because not many people can work on neutrophils. Because the work that we did on neutrophils probably was one of the very first neutrophil work in the field of HIV after 40 years. Because neutrophils, you cannot freeze them. You cannot keep them. You have to work on them fresh, right away. So we had this opportunity to go to the clinic, bleed patients with the help of Dr. Stan Houston, and then come to the lab and work on the neutrophil. So John Walker, which obviously at the CCI and with the clinical trial, and Dr. Slieger and Osman with the COVID studies, and obviously the funding sources, CIHR, has been privileged to be supported by CIHR so far, and Alberta Cancer Foundation and Glycomics, and also at the start of the COVID, I was lucky to get a kind of the global competition. I got a FAST grant for COVID research.